Hello everyone and welcome to Twist Bioscience's workshop at ESHG 2021. My name is Chris Thorne and I'm Senior Manager Field Marketing at Twist. This year we can't be with you in person, but we're delighted you can join us virtually. And I'm very pleased today to welcome our speaker, Kristi Kruusmaa, who is the Head of Research at Universal Diagnostics. Kristi has a background in genetics and biochemistry and has spent over 10 years working in the biotechnology industry in areas such as cancer biology, liquid biopsy, method development and data analysis. At Universal Diagnostics, she is currently leading the work of a multidisciplinary team in diagnostic product development, with their main focus being on gastrointestinal tract cancers. Today, Kristi will be discussing their use of Twist's target capture technology for accurate colorectal neoplasia detection through targeted bisulfite sequencing analysis of cell-free circulating tumor DNA. Thank you, Kristi.
Hello everyone. So first of all, I would like to thank Twist Bioscience and the conference organizers for giving me an opportunity to give this talk. So the title of my talk is Accurate Colorectal Neoplasia Detection through Targeted Bisulfite Sequencing Analysis of Cell-Free Circulating Tumor DNA. This is my disclosure, and I would like to divide this talk in three parts to talk about colorectal cancer problem, the solution that we have in Universal Diagnostics, and further applications of the solution. So, first of all, I think all of us are aware that cancer is a major problem worldwide, and colorectal cancer, out of all the cancer types, is ranking quite high in all parts of the world. The same goes for the mortality rates, which, despite all the efforts over the last decades, are still low. The mortalities are high, and the 5-year survival rates are still quite low, ranging from 60 to 65%. So what's crucial for the colorectal cancer topic is the early detection. So if the cancer is detected in localized stage and the survival rates are up to 91%, whereas when detected in distant and advanced stage, it's only 11%. Unfortunately, currently only about 37% of the cancers are detected in localized stage. Even more, there is an important additional stage in the whole progress of cancer development, colorectal cancer development, and that is the advanced adenoma stage. So 85% of the colorectal cancers are progressing from large polyps or high dysplasia adenomas. And so that would be the sweet spot for actual early detection, because if you manage to detect the patients with large polyps or advanced adenomas, then you would actually be able to prevent cancer from ever detecting, because you could be removing those lesions through colonoscopy. Unfortunately, the existing screening tests that are available today have certain drawbacks. So colonoscopy itself is quite invasive, and sending all the patients to colonoscopy is expensive and could be also life-threatening for older patients, as well as doing this too often would be causing a lot of inflammatory problems. The other option is fecal testing, which is quite unpleasant for the patients, and so the compliance rates are remaining quite low for the screening population. So that's where the liquid biopsy comes into play, as blood testing is easy to obtain. You can do it in your regular GP's office. It's relatively non-invasive. So the adherence rates are a lot higher than for other screening methods. You would get the full pathological profile of the patient at the moment of testing. You can detect different biomarkers, and these signals are tissue-specific and would help to determine the actual condition of the patient. So that's where Universal Diagnostics comes into play, and we are working on blood-based solutions for early detection of CRC, and the biomarker targets that we are mainly looking at are epigenetics biomarkers, first-hand methylation markers, because they are highly disease and tissue specific. They appear early in the cancer development, and it's quite robust to measure them in clinical lab environment. And of course, the point that you can detect them from blood is important. Further on, the methylated CpG islands correlate with also constant and close chromatin structures. So measuring the chromatin structures and from there off the cell-free DNA fragmentation signals is an additional marker type that can be incorporated into analysis. So in our case, we have determined that the methylation signals as well as open chromatin region profiles are distinctly different between the colorectal cancer patients and control patients, as well as between the different tissue types and patient types. So this kind of markers are really the ideal markers for early detection. So the key for going for biomarker selection is, of course, starting from the disease itself, so analyzing the tissue from the cancer patients, so cancer tissue as well as control tissue. In parallel, determining what are the potential biomarker regions, determining what is the cancer-specific pattern in there, then also filtering these signals against puffy coat that could be causing problems in blood analysis, filtering against healthy cell-free DNA, as well as other cancers and other control tissues. And then from that point on, the key is to transfer these targets or capture these targets in plot-based assays, and for that going into targeted sequencing in plasma. So for this purpose, we have collaborated now with Twist Bioscience, who has allowed us through their methodology to capture and design the probes for our targets of interest, the regions of interest. The key in here is that for methylation analysis, you need to do the conversion of the DNA to be able to detect between the unmethylated and methylated reads. So the Twist Biosciences probe design is specific to the unmethylated as well as methylated regions in a converted setting, and it's appropriate for low input analysis, such as cell-free DNA analysis, of course, is. So the chemistry itself is easy to incorporate into any converted library setting. So the prep library fragments are blocked for any kind of unspecific binding, then hybridized, the probes are hybridized and then captured. And in our current UDX methylation panel, we are covering more than 600,000 base pairs, and we achieved more than 4,000 times enrichment for those targets, and 85% of the reads are actually on-page. So it's a very specific, very, very accurate way of capturing the targets and enriching for it. And the performance is reproducible using only 10 nanograms or even less of cell-free DNA. So this kind of approach has been used in our hands for developing our model for early colorectal cancer detection. So we have our targets, we had our panel, and by doing the analysis on 4 milliliters of plasma, we had a patient cohort of 263 patients that was separated into training set and validation set. So in a training set, we had matching controls in colorectal cancer patients, matched by BMI, age, gender, origin, also the cancer location, proximal and distal colon. And validation set then included colonoscopy negative controls, hyperplastic polyp patients, non-advanced adenoma patients, previously undiagnosed gastrointestinal disease patients, and colorectal cancer patients from 3 populations. So the process of analyzing the plasma is based on the Bisulfite conversion, library preparation, and hybrid capture with Twist probes, and then sequencing, upon which we apply our AMBER score. So this is read-based methylation signal scoring for each region and each patient. Then this information is combined into machine learning model. So the model was built on the training set and then applied to the validation set for the prediction. With such setting, we have managed to achieve a really outstanding results in terms of the colorectal cancer sensitivity and specificity. So already on the stage 1 level, we had 83% sensitivity, and this was increasing to 100% for stage 4. The overall sensitivity was 92%, and the specificity was high at 97%. And the sensitivity is comparable for proximal and distal cancers, meaning that the regions that we have, the model we have developed is equally efficient for right and left-sided cancer detection. The specificity in subgroups of colonoscopy negative, hyperplastic polyp, non-advanced adenoma, and newly diagnosed gastrointestinal disease patients was in all cases more than 90%, actually more than 94-95%. And it's that the specificity and the signal itself is not age dependent. So regardless of the age group we looked at, the specificity is still over 90%, showing that the signal we see is really specific. It's very sensitive for the cancer itself and is not relying on any kind of co-founding parameters. So the individual markers that we have been looking at are connected to the pathways in cancer and signaling pathways. And so the pathway analysis really shows the link to cancer and cancer progression itself. One of the main markers that we are looking at is FGF14, for example, and this has been shown to be a tumor suppressor. So not only are we detecting methylation regions, but also trying to put these into a biological context and then see further if we see any potential for targeted therapies or any kind of other functionality in here. So going further from colorectal cancer, then we have also had a study on advanced adenomas where we were checking on control patients, so 136 control patients, and then advanced adenoma patients from different histological subtypes as well as dysplasia grades, dividing this into cross-validation setting in here, and based on the similar approach of looking at the AMBER score or the read-based methylation signal scores and building a model and validation in here, we managed to get as high sensitivity levels as 55% for the advanced adenomas at 90% specificity. More importantly, the sensitivity for the high grade and carcinoma in situ patients, which are the most advanced adenomas and most severe cases, we managed to get 63% sensitivity, which is really high. And this is a very important part for cancer prevention, as already mentioned. Also in histological types, the 60% for serrated adenomas is really important mark, as serrated adenomas are, and with dysplasia, are really aggressive and have more potential for colorectal cancer development. So looking into the pathways again of the markers that are in the panel, then connections are made into cancer pathways and regulatory signal pathways. So once again, the biological link is quite obvious in here. So what are the further applications of such an approach? So also, I already mentioned the open chromatin regions, so combining this information with the nucleosomal positioning and methylation analysis, we could even enhance the signal further. So we have done some preliminary analysis on this, and you can see in the PCA below here how the separation between the control group and advanced adenoma and CRC group is quite clear in here. We are combining this now together into targeted analysis approach with twist probes. So combining this information, trying to also enforce this into the biological context, so making connections between the methylation regions that we have and the open chromatin regions that we have, and trying to explain the functionality further as well. So not only developing a screening test, but also trying to understand the biology behind this. Further on, we are going further from colorectal cancer to other gastrointestinal cancers. So here are a couple of regions for pancreatic cancer and gastric cancer. On the left side, pancreatic cancer case, the clearly hypomethylated region can be seen in comparison to the Buffy code, other pathologies, and sulfur DNA. Whereas in the gastric cancer case, you can see a hypermethylation for the red line for the gastric cancer patients. So we are hoping to combine all of this together for a multi-cancer test and together with a targeted hybrid capture method to have an assay that would be able to detect not only one cancer but multiple cancers and with a high accuracy. We have been doing this work mostly on Bisulfite converted DNA. However, we have been comparing this to enzymatic conversion as well. And then on this slide, you can see that the values for sequencing quality on replicate samples are quite comparable in terms of the initial reads and optical duplicates, deduplicated analysis, and filtered analysis. So either one of these could be used. Also, the base quality is quite comparable on page rates and duplications. And if you're looking at the BCA on this replicate analysis, we had 8 samples that were control samples and 8 samples that were CRC samples prepared with 2 methods. Then on the BCA, you can see that the separation in here is based on the disease state, not by the preparation state itself. So showing that the signal that we see is really disease specific, and both methods are working for this. So key takeaways in here are that the highly multiplexed targeted hybrid capture methylation analysis can be used for efficient enrichment of the targets on sulfur DNA. Well, combining this into signal calling and machine learning approaches, we could achieve really high sensitivity for the early detection of CRC at stage 1, 83%, as well as an advanced adenomas at 55%. And this method can be, of course, further expanded into methylation and open chromatin region capture, extended to multi-cancer detection, and it is flexible method to be used with enzymatic and Bisulfite conversion. And with this, I would like to thank all of our collaborators from the technical side, so Twist Bioscience, Diagonal, Gene Canada, and UPC, and also all of our clinical associates from different countries, from Spain, from Germany, and from Ukraine. And with that, I would like to thank you, and I'm open for any questions.